Just a couple of intro slides about Finavista. Like I mentioned, or Asesha mentioned too, Tony and I formed the company back in 2014, about this time in 2014, and yeah, we've been growing. We're completely services based. We completely focus on high content, so image based analyses, primarily using the Cellpainting systems, and we've been growing rapidly over, especially this year. As I'm sure a lot of people in the audience know, high content's a very versatile platform technology. You can use it for loads of things, not just sort of screening. In fact, the majority of the work that we do for our clients is in more sort of fundamental, trying to understand the mechanism of action for some of their therapeutic agents, whether they're small molecules or biologicals or anything else, but trying to sort of understand what's going on with the biology. Just a little plug for our friends who are hosting this. When we set up the business Finavista, obviously there are lots of other, not lots, but there's several other leading providers of high content systems, and maybe for sure there's some bias, in sort of the past history that we've enjoyed with Thermo, so that makes it easier to go with something But there's some real strengths with the Thermo system, and I think these are three that came to mind. As a small business, we're at 25 people now, cash flow driven. We can't have instruments that are down for a month or down for 2 months. That's going to be really, really bad. Of course, one of the key things with the Cellpainting so design has always been the integration with informatics, and so that's really useful in terms of getting fast turnaround on projects and getting not just fast turnaround when things are going well, but fast turnaround when you're trying to troubleshoot, as is often the case. Trying to figure out what's going on. This assay window is not the way it should be. This antibody is not behaving the way it has in the past. So you want to get a fast answer on that, so you can get back to your clients as soon as possible. And then also return on investment, not necessarily region of interest for those. Image segmentation joke there, early days. Anyway, so yeah, I mean, the Thermo systems are really capable, and they're not the priciest options out there. There are definitely systems out there that are cost 4 or 5 times the instrument cost, and so getting something in that's dependable. Kind of, I always use the analogy of being like a Ford F-150. So getting something that's dependable and reliable. Just summarized here. So then just final couple of slides here. So this is kind of what we do. Obviously, imaging-based assays, you can do tons of stuff. 2D fixed cell analyses, time-resolved analyses, co-cultures, more and more 3D analyses of organoids and spheroids. Also, microfluidic systems from providers such as Inspiro and the Metas emulate these companies. And what we typically do is handle from the design of the experiments with our clients through a very sort of collaborative style of engagement, come up with a plausible plan, and then execute that plan from growing the cells, treating the cells, fixing, staining, imaging, putting together everything for the clients. Things on the right, we can offer those, but there are a lot of other really good providers that do that, so we don't typically lead with that. Broadly speaking, the kind of services we offer can be broken into 3 categories that may or may not lead on to each other. So a lot of it will be, can you guys develop an assay for this? It'll be the feasibility section. After that, or people might come to us with something that they already know you can develop an assay for. They've done it in a kind of a manual, maybe confocal mode in their imaging facility. And they want to say, can we scale this up? So we'd come in and help them with that. Other people might have come through these first 2 stages and say, okay, we're happy with what we have now. We want to start routinely sending you, 10,000 compounds a month. Can you screen through them and send us the data on a regular schedule? Other people might come to us with an assay they've developed in-house and say, you know what, we don't want to do this in-house anymore. It's taking up too much time on our instruments. Here's the protocol. Can you guys do it? And then essentially we can come in and out of this process at any part. So we can transfer back the assay. Definitely a lot of our clients in the big pharma space have far greater capabilities, especially in the screening side of things, for doing 1 million compounds a week or something like that. That's not where we are strongest. Okay, so this is just, I think this is the final slide on the background about us. So we're a very PhD heavy company. We're just expanded our lab space. We're at 7,500 square feet. We're located near UCSD, just north of San Diego. A lot of tissue culture, a lot of liquid handling inside hoods. As you can imagine, a lot of compute infrastructure to support all this imaging, including some downstream software packages from Olympus, Amaris, or Huygens, things like that. So we really try and get as much out of Celomics and high content as possible. On the right-hand side of the slide, you can kind of see a list of some of the relevant hardware and kind of in general what we use it for. So as you can see, we're pretty heavy there on the thermo Celomic systems, and we also have a GE system just to keep things spicy, and then we also have a IncuSite. We have a Olympus confocal. We use microfluidics, and two systems shown at the bottom there. The one highlighted in red is our newest addition. It's this isoplexis instrument, which is not actually high content, but is kind of nice nice technology that complements high content. Yeah, have a look at that. And then the big box. This is not shown to scale. The instrument on the right there is this really cool. Basically, it's like a spheroid handling instrument from Yamaha motors that we've been really happy to collaborate with them on developing and developing applications. So that's really an integral part of our ability to handle organoids and spheroids at scale. And I'll have a couple of slides on that later. Okay, so why use high content? Why not use flow cytometry? So as Sesha mentioned in the intro there, Dave Gebhard, who was at Pfizer for a long time and had a background in flow cytometry, he and I were in the same training course together, the HCS 101, back in 2001. And Dave came with a really strong background in flow cytometry. Flow cytometry is still a very relevant technology and has real strengths over high content. But this slide's just really supposed to highlight where high content steps away from flow in terms of its capabilities, and particularly in where you want to answer questions about where a marker is in terms of subcellular localization, or in the context of cell morphology, in cell-cell interactions, and of course kinetic measurements in terms of migration and then also 3D complex structure. So that's where high content really is the way to go. Okay, so getting to the main part of the talk now, just going to go through so 4 broad areas here. I'm going to start with an example talking about mitochondria and looking at mitochondrial health. It's always, no matter what the biology that we're looking at or the sort of disease focus, mitochondria are always coming up as something that people are interested in. And so just got an example here of how we tackled that for a client a few years ago. So this is using, for those who are familiar with the Celomics HCS Studio, I think is what it's called now, but the analysis software. Look, that's popped up. So this is using spot detector. a really flexible, capable application. And the challenge here was to look at this structural marker, with COX-4 antibody staining, and determine the effect of treatments on the degree of fragmentation within of the mitochondria. And as you can tell just from looking at the images there, the cells are very, each cell is different. I'm going to see if my marker will come on here. There's a pointer. Hopefully you guys can see this, but depending on the size of the cell, the presentation of the mitochondria is very different. So the degree of punctate structures is going to be different depending on the size of the cell. So long story short, what we did here was using spot detector and various thresholds. We analyzed this image 3 times using different slightly different segmentation approach, and then filtering. So that the yellow panel here shows kind of the total mitochondria per cell, and the 2 panels down the bottom here use the metadata associated with each of those yellow objects, or derivatives of those yellow objects, to put them in a bin of being sort of the healthy elongated mitochondria or the small punctate mitochondria. And that way we can use these 3 compartments to come up with a kind of a ratio to get a kind of a normalized distribution of the amount of punctate structures, mitochondrial structures, as a function of the sort of the total mitochondria in the cell. Okay, and we could go a step further than that. So of course that's just a structural aspect. As you're probably aware, there are a lot of different functional markers for mitochondrial health too, that are typically fluorescent when the mitochondria have a membrane potential, and then lose that fluorescence when the mitochondria are perturbed. And so we were able to combine that too, so not just a structural analysis, but then add a functional component to it there. So you can see here in these nice healthy mitochondria, you have a relatively bright signal for the mitochondria. And that as you increase the amount of this decoupling agent, CCCP, you lose that intensity from the mitochondria, and the mitochondria become more punctate. So you can combine those, that structure and functional aspect. And one of the things that we did for our client in this case was to combine these metrics into a single axis so that you could rank all the compounds. This is particularly useful in this case for the chemistry folks who wanted to just rank all the compounds. For biology members of the team, they maybe wanted to sort of break the data apart. Okay, let's look more carefully at structural only versus functional only differences, and so you have that ability too. But so I guess the long story short here for this mitochondrial aspect is, using this Lomics platform creatively, and then also getting the most out of the data that comes out of this Lomics platform in terms of I'm trying to think of the right word, sort of crafting it so that it's presented to the client in the most useful manner. Okay, any questions so far on that? I'm just saying that so I have an excuse to have a sip of coffee. Doesn't look like it. Okay, so now we're going to move into the big category here. This one's going to take a while to get through, but this is kind of a mishmash of immunology, immuno-oncology, and oncology. Kind of just a bit of all the tricky little cells that are hard to work with. So talk about that a little bit. So as I mentioned a few slides back, one of the interesting things you can do with imaging that is more difficult to do with flow is determine the localization of a marker within a cell. And here we have an activator of T cells. So actually backing up a little, one of the challenges in high content is using non-adherent cells. So, T cells and primary T cells and PBMCs and things like that aren't particularly well suited to high content in the first place. They're not like HeLa cells that can grow in a 2D culture and they behave. So, coming up with methods to handle these cells and get them into an imaging compliant format. I should mention the majority of all of our assays are performed in 384 well plates, so they're low volume and quite scalable. And so being able to do that with these kind of cells is not trivial, but it's something that actually Tony, my business partner, spent a lot of time early on in the company coming up with methods to do that, and now the whole company does this routinely. Once you've got that in place and you've got these weakly adherent or non-adherent cells distributed and attached to these imaging plates, then you can do a lot of stuff. You can look at cell-cell interactions. You can look at in this case, you can look at subcellular localization. Here's a question from Pam. Oh, which mitotracker do you use for the membrane potential dye? It's Mitotracker CMX Ross is the one that we use, and then magnification for the mitochondrial stuff. That was I want to say the images that we're looking at there were 20x. It was kind of a trade-off between 20x and 40x, depending on the cell type, how big they are, and getting enough measurements, but typically 20 or 40x. Sorry. Anyway, back to this. Yeah, so here's just a zoom in here of these T cells, and hopefully you can see the difference there between the localization in yellow with the yellow arrows where the Nfat has translocated to the nucleus, and in the sort of pinky purple color, it's still excluded from the nucleus. What's the value of this? Well, what's interesting is that Nfat translocates very quickly relative to more conventional markers like CD69, which is a kind of an overall expression level marker, which peaks at around 2 hours, as shown here on the right, with the black trace, the black plot. So if you're interested in finding acute activation, Nfat is a good one to go with, but it's not something you can necessarily do with flow cytometry. It's just an example of where high content for all the extra effort, what you get for it. Scroll down here, make sure I'm not missing any other questions. Another question. Oh, sorry, one more question from Alex on mitochondrial health. Applicable to different tissues for sure. Yeah, I mean, so that was just looking at I think it was just HeLa cells in that example, but we've taken that approach for human iPS-derived cardiomyocytes. I think even later in this presentation, I'll have some examples using human iPS-derived neurons, albeit in that case, the way the cells are present, the shape of the neuron makes it more difficult to get those structural measurements, but the idea of looking at structure and function. But yeah, generally speaking, it can be pretty versatile and apply to a lot of different cell types, as long as you kind of have reasonable expectations in terms of what you're hoping to get from it, based on the cell shape. For example, I wouldn't try and do it on primary B cells that are just so small and have such a small cytoplasm. That would give me even more gray hair than I have. Okay, so here's an example of looking at cell-cell interactions for these type of PBMCs and these actually blood-derived dendritic cells in co-culture with CD4 positive T cells. And what we're able to do there is look at the proximity of neighboring cells and count the incidence of immune synapse formation, which you can see highlighted with LFA1 with the green. So that's something that's really only doable with imaging. Oops. And similarly, on the sort of the the punctate idea, looking at inflammasomes too, looking for the incidence of these punctate structures, whether using ASC or NLRP3. But then again, with imaging, not only can you kind of get a binary sort of yes, it's there, or no, it isn't, or but you can actually start looking at the distribution, as is shown here on the right. You can start seeing, well, some of these cells appear to have more than one puncta per cell, and so you can start sort of categorizing those cells. Other measurements too, you can look at the intensity if that's relevant, the size, the shape, and things like that. So again, these are really this is where the high content aspect of high content screening or analysis really comes in. Okay, now we're getting taken up a level, mixing up the cells. Makes it sound a lot easier than it is, but here we have, a solid tumor cell line that's co-cultured with the Tcell fraction, and we can look at killing of that target cell line pretty easily by staining it beforehand for mixing, and then quantifying the loss of those cells if there is a loss. We can also measure the Tcell fraction. We can look at death of both populations with DRAC7. Of course, we can add in any other antibody-based markers that are relevant too. But the thing I wanted to highlight here was the ability to, you know what, there should be a thing, but I think you can pick up on it. At the bottom here, you can see that the target cells are being decorated by the T cells in kind of like a swarming or a clustering approach, and that's something that's high content and is able to actually quantify. So if in this case you don't see a massive loss of the cell lines, and they're not being completely wiped out, but you get the hint that, well, something's going on. The T cells, maybe they're not working as quickly as we'd hope, but they seem to be at least homing in on the target cells. So that's some extra potential information. You can kind of see that here. You can pick up on that, hopefully. Okay, so now taking it up another level, combining 3D culture. So this is actually a slide from our hosts today, Thermo Fisher from Chris Langsdorff, a poster that he presented a couple of years ago. And here we're, basically, instead of culturing the target cells in a 2D format, you're pre-culturing them in 3D, and then adding, in this case, natural killer cells, and looking at infiltration and death. But so same concept, but in a 3D format. And again, that's something that you can easily transition with the same markers and the same imaging platform, and get corresponding, see if there are differences in sort of the therapeutic approach or efficacy as you move from 2D to 3D. So one of the things I'm really proud that Vista has been involved with over the last year, or just about a year, is a partnership with Champions Oncology. Champions has a huge repository of PDX tissues, and these are PDX, not PDOX. It's a slightly different technology. I should just be clear that we use PDX, at Vista. But so Champions provides those to us for ex vivo companion studies that go along with their many studies they do in vivo. But it kind of builds off of the last slide that we saw from Chris Langsdorff's work. So here's just an idea of how those PDX fragments look in the 2D culture. There's a whole kind of distribution of sizes and shapes, and this also varies wildly based on the type of tumor that you're working with. Scrolling down on questions here. Okay. So here's a kind of a eye candy view of that, of one of these. I think this is actually a pancreatic tumor model. And you can see the Herc staining top left, you can see a cell tracker, you can see the dead cells labeled DRAC7. And kind of similar to the 2D approach, you can go through and quantify the number of total cells, the fraction that are viable, and the fraction that are non-viable. Cell tracker, green in this case, is typically only, I believe, esterified and retained within the cell if the cell's viable. So as you can kind of see just by eyeballing it, you don't really see many examples where the cell is DRAC7 positive and cell tracker positive, although, you know, obviously in this 3D example, there is some overlapping, which complicates it a little bit, but that's the model at least. Here are some real life examples of how those PDX models look, and kind of a cartoon of what we think is happening on the left. Yeah, it's pretty challenging. It's quite a complex, as you can see. Presentation of the biology is not, it's not just like a 2D culture. So it's there's a lot of procedural challenges in getting samples consistent and getting the data to a high quality. Here's kind of a drill down into an example here. And I hope you can see there the, you know, obviously this is you know, you don't show the bad examples, but this is a pretty black and white example where you see an increase in the number of dead cells treated with this conveniently named drug A. But yeah, it's all quantifiable and gives a nice dose-dependent calculation. Oh, we have a question here. Have you tried to image this with the new Eureka Scan function, given the seeding variability? I have not. I do not know what that is, Nicholas, and I would love to. This question from one of our hosts, Nick Radio. I'm sure most of you know, but yeah, I haven't tried that. I'll be interested to learn more about it. Okay, so any other questions as we move into the kind of the 3D? We're kind of in the home stretch now. So we're going to talk a little bit more about the 3D aspect of things you can do with high content.
I think, James, just to mention before we move into the 3D models, so the Eureka Scan function is a new function for our platform that allows to do essentially a double pass. So you can screen at lower magnification, find the tumor, and then zoom in at the higher magnification. The instrument can do that in automated fashion. So it's a very useful tool for those customers that are doing 3D scans.
Yeah. Oh, thank you for. I didn't know that's what it was called.
Yes.
I didn't know it had a name. It must be real. Thanks for explaining that, Victoria. Yeah. So we do use that for things like this, the stuff on the screen right now, where you'll have like one big structure within a well, and you don't want to scan the whole well. You want to find where it is. Funnily enough, one of our ex-employees, a really talented guy called Javier Pineda, who worked for us, he had built his own version of Eureka Scan using using like just sort of serial scanning, and it was really it was a pain, but it worked, and we needed it to work. So we were really glad when you guys came out with an integrated option for that. But yeah, so this slide here is, I believe, of a StemOnyx microbrain, and it's to highlight the importance of clearing, so tissue clearing. So this is where things get really non-fun in moving from 2D to big 3D, big dense 3D structures, is the importance of clearing the structures so that reagents, antibodies can penetrate, but also so that the light can make it back out of the structure. And you can see in the top, without clearing, you really get only superficial staining, whereas with clearing, you get a lot better penetrance. And of course, there are lots of different reagents sold commercially for clearing. There are lots of different methodologies, but they tend to last days to weeks They're really laborious, and there's a link here to, or just pull out to cluster now from Tcell in 2019 here for a review. And the nice thing is that there are really well documented, published protocols and reagents that you can make yourself, and we've made gallons of the stuff, and yeah, so it works really well. So what you can do with this, so this is a little plug here for our friends at Yamaha Motors, their cell handler instrument. It's an incredible piece of equipment. It's actually born out of semiconductor technology for doing printing. And on the left down here, you can see it has these like kind of inverted pyramids that have a hole in the bottom, so that structures that are greater than, I think, 30 microns in diameter will get sort of caught there, whereas single cells will fall through. And then you can go through and pluck them out and transfer them to an imaging plate. And that sort of plucking out can be guided not just based on size, but also shape and fluorescence. So you can do some real sort of quality improvement in terms of generating your assay plates or imaging plates. So once you've done that, you can go through and do a lot of profiling with different markers of interest, or develop what we call palettes or combinations of markers into groups of 3, 4, 5, something like that. That makes sense. So it's a really great enabling technology. Actually, what's really cool too is that that system will not only pluck from that inverted pyramid plate, which is called it is a plate made by Yamaha, but you can also embed the in this case intestinal organoids into a 3D gel, and it will be able to go through and pluck them from those gels, which is super cool. And you can see here before and after. Here's another look at these intestinal organoids actually made by Definigen. I think they're a British company. Don't hold that against them. Yeah, so you can go there, and you can see they have a really intricate structure, which if we zoom in on, we can get a sense of the lumen. So it mimics the intestinal lumen and the microvilli invaginations that come in there. So you get a real tissue structure, and this is actually stained here using kind of a derivative of the sort of cell painting kind of approach. Again, this is actually back to the mnemonics microbrain samples here. They're really dense structures, and they have a combination of neurons and glia, and you're able to look at the distribution of those two or more fractions, and quantify those distributions and any potential treatments, how it's affecting the health of those different subpopulations. The question here again from Nick about depth of imaging with the 3D and clearing. It definitely depends on the labels and the density of the samples. Denser samples are going to be less permissive for deep imaging, but and then of course you want to make sure you have the right sort of optical setup in terms of magnification and working distance and so on. But several hundred microns is doable. Not at super high magnification, not so much, but if you make a good trade-off between magnification, working distance, and you have the right sample and labeling, you can definitely do a few hundred microns in depth. Okay, last section here, and we are running out of time. This will be fairly fast. So 2D, looking at neurite outgrowth and neurite health. There's lots of different ways you can do this. the Cellpainting platform has a neurite health profiling algorithm that's kind of a pre-configured for doing this kind of thing. We use that, especially when we want to measure branching. But you can also use spot detector. In this case, the masks here are using spot detector, and I think you'll agree that they work pretty well at sort of finding all the relevant structures. So again, you know, using all the tools that are available, you know, creatively, I think is definitely worked for us. Again, here, this is essentially very similar to the previous slide, but is a short-term culture where we look at the sort of the neurite outgrowth during the first few hours after plating. These are all human iPS-derived neurons from Fujifilm, but we can do this with client-provided cells, to some degree, some primary cells too. Things that's obviously cell lines too. So it's pretty versatile. You know, here we can go through and again using spot detector, we can measure the amount of Tujuan staining and the neurite marker over time, and look at the and get a really large assay window between negative and positive control. Then you can start layering in more complexity in terms of markers. You can look at apoptotic indicators. You can obviously look at you know, how many dead cells you're getting. What's interesting is the DRAC7 marker. In this particular cell type, again, human iPS-derived neurons, the morphology of the hook staining for dead cells is very obvious, such to the point that the DRAC7 label really isn't even necessary. You can do it almost completely using the morphology features of the Cellpainting software just to identify the reduced area and the increased brightness, and a very useful output feature called variation in Tcell bar in 10 channel 1, which will typically identify these rounded up nuclei very easily, which means you can free up the DRAC7 channel for something else. Other indications of cell health, as I mentioned earlier, way back at the beginning of this talk, using MitoTracker CMX-ROS as an indicator of mitochondrial health, pairing that up with autophagy markers like LC3B and LC3AB, but antibody-based markers. There are also engineered fluorescent protein constructs that can be used too, and some of our clients have cell lines that they've licensed and we can use on their behalf too. But certainly you can start combining up relevant health features such as these. Here's a kind of a bit more in-depth look at that, the sort of neuronal equivalent of the mitochondrial health assay that we started off with. So, you know, first thing is, you know, the structural, sort of the presentation or the appearance of the mitochondria in neurons is not as sort of aesthetically pleasing as you get in, you know, for example, an epithelial cell. What's interesting is, you know, we were always concerned that it was as a matter of the the magnification, and so, you know, we looked at these neurons at a much higher mag and 100x magnification, so on different platforms, and from the best of our sort of appraisal of this, it really does look like the mitochondria in these neurons, at least, tend to be much more punctate and less reticular than you see in epithelial cells, just you know, as a sort of a baseline. So, but anyway, the approach you know, stands up, although, you know, it's not quite as sophisticated as where you're looking at the elongated structures, but you can still get relevant information on the sort of structure function dynamic between you know, using this approach. Structural marker. So there's a few different structural markers. There's a question here about what's the structural marker for mitochondria. COX-4 or TOM20. Those are two that work really well. So I think we're going to the end here. This is this is pushing the envelope as far as what you can do with this is using the co-localization application within HCS Studio. And what we're doing here is a three-way co-localization between the pre and post-synaptic markers, and so shown here on the left and right, in the middle and the right. But they're sort of gated or masked by this blue marker, which is the Tujuan staining. So basically, you're looking for the number of objects, synaptic puncta, that are in proximity to each other, but under this blue mask. And here's a real, sort of more real life example of that. Yeah, so basically it works pretty well. The staining, so Tujuan, as shown here in purple, will give you some puncta that does get the sort of proximal end or the terminal ends of the microtubule stacks. You tend to get sort of accumulation of staining. But you can go through and quantify in a 384 well plate. You might quantify 5 million puncta, and it works. And what's interesting with this assay is the ability to measure changes in kind of all the different axes of the assay. So you've got obviously neuron health. You've got the network you can measure, the Tujuan network. You can measure the number of puncta, the expression levels of the pre and post synaptic components, and then of course the co-localization. So all of those different outputs can be measured independently. And what we've seen is that they do vary independently too, so it can be quite interesting to sort of tease apart the effects of different therapeutic agents or growth conditions even on those different aspects. And so actually, then you can go a step further and start doing co-cultures of, in this case, glutaminergic neurons with astrocytes, glia, all human iPS derived, all from Fujifilm, and look at the buffering effect of astrocytes in response to a large insult of glutamate and glutamate toxicity. But you can also look at synaptogenesis too in this context. Here's just the data. Let's skip through that. Last couple of slides here. As I mentioned briefly before, cell painting is a very hot topic, particularly amongst our big pharma clients. Something you can do with the Celomics platform, even if you don't use Cell Profiler as your analysis suite, you can use the dyes. And one of the nice things with the CX7 is the fact that it has that fifth channel, the 750 channel, which not a lot of other instruments support of this kind. So it does give you the option of doing a full cell painting palette and then putting an antibody of choice in there to sort of do follow-up studies and sort of see the correlation in the world of cell painting where your particular marker may reside. You can use the old machine learning, AI, deep learning, whatever you have in your bag of tricks, and try and make sense of all that data and see what comes to the top. And definitely spend some time doing that on behalf of our clients. You can also build your own cell painting approaches where you take these antibodies that we use routinely, or check and develop new markers that you might be interested in, and throw them into your mix and sort of basically come up with your own kind of cell painting approach. So that's definitely something we've had good success in. And those again can be general, neuronal, they can be functional markers too. Here's a case of where we did this. Look at different mutant lines in TDP43. We can go through and quantify them and do some PCA in this approach to sort of cluster the different genotypes together. And there we go. We have a few minutes left for any questions that might come up. Again, thanks for your time. To mention all the people that did the work and our clients that support us and so on. But we're happy to answer any questions that people might have.
We have 6 minutes for Q&A.
Thanks, James. Really appreciate it. There is a question from Pam Swain on what is the structural marker for mitochondria?
Mm-hmm. Yeah, so yeah, COX-4 and TOM20 are ones we use routinely. They're published markers. There's good antibody, commercially available antibodies for those, and they work really reliably.
That's awesome. Any other questions from the participants, from the attendees? James, I have a question around where the customer segmentation is that you see. Where are the asks usually? Is it neurobiology? Is it 3D, immuno-oncology? Where do customers come back to you and say, hey, I'm struggling with this assay. Can you help me out?
Yeah, so it's pretty broad. I mean, for sure, there are certain things that kind of lend themselves, certain applications that lend themselves very well to imaging, particularly, neurobiology and CNS. So definitely when Finavista was starting out, more than 50% of our business was in that space. As things have sort of matured and immuno-oncology is definitely come up to sort of the same level of CNS over the last 2 or 3 years. But I think the one thing, the sort of recurring theme that clients come to us with is not really one particular problem, but in juggling all the problems. So that's really what we try and bring to our clients is, it's not just an imaging problem. It's not just an informatics problem. You've got to kind of know what you're doing in terms of setting up the assay. You've got to know what you're doing in terms of, the cells have to be healthy. They have to be placed at the right density. You have to have good antibody. You have to have everything. And that's really what we try and do for our clients is, bring everything together and make sure that, the whole system is working cohesively.
Yeah, that's awesome. Johnny Wang has a question on, does the CX7 has any specific analysis panel for cell painting? I know you mentioned the advantage CX7 has obviously is the near IR capability, right? Does that make it as a standalone advantage and make it more amenable to cell painting per se?
Well, so one of the things with cell painting, and the originators of cell painting, that we had a panel discussion at FCI2 a couple of years ago, and Anne Carpenter's ex-colleague of mine from Whitehead, and has been very upfront about, and her collaborators been very upfront about, the design of cell painting, and that, it's not set in stone. And, you can adapt it to your more specific needs. And also, the dyes that are used bleed through into adjacent channels. And depending on the cell type you use, they're going to have different degrees of labeling. For example, as you can imagine, tolloidin is going to label a HeLa cell very differently from how it labels a cardiomyocyte. Cardiomyocyte is going to be loaded with actin. So, exposure settings, your analysis settings are going to be wildly different between those cell types. So I think the way a convoluted answer, but the nice thing with the CX7 platform, the near infrared, is it gives you a bit more versatility in that you've got more literal spectral space to play in. You've got the extra channel, and if you want to separate some dyes, to make sure there's no bleed through, you can do it. Or if you want to layer in an extra antibody, you can do. But particularly to Johnny's question, there isn't a specific sort of canned algorithm, but that might be something that someone might consider in the future. I think it'd be great. A canned algorithm for doing cell painting style analyses. We tend to use a sort of combination of morphology explorer, spot detector, and departmental analysis, things like that. But you can get a lot done with spot detector.
That's awesome. Again, if there are no additional questions, I just wanted to thank James again. This was a wonderful talk. I think we can take this offline. The only question I would probably have, James, is has anybody asked you about 3D analysis? But we can have a separate conversation around that.
Oh, yeah.
Yeah, I just wanted to add to James. Thank you so much for sharing today your work, and I think it's very exciting what you're doing in collaboration with Champions Oncology in those PDX models. But I think we all know the 3D models can be very challenging, right? So I think this is kind of a nice segue to our next talk tomorrow with our product manager, Nick Radio. So he's going to talk specifically about some of the new features that can help us get through those challenges with the 3D cell model. So hopefully we can have more people join us tomorrow to hear about how Eureka Scan can help and the new 3D analysis capabilities through Amira.
Great, I'll be there.
And the slide you see there is the title for his talk, Investigating Three-Dimensional Life Sciences Biologies for High Content. So please do tune in tomorrow. It would be exciting to hear the updates that Nick has around 3D biology as well. So that's awesome. Thanks again, James. Really
Thanks, everyone. Have a good one. Bye-bye.
Thanks.
Thank you.
Bye.